All right. Okay, welcome those of you who are watching after the fact. We're gonna go ahead and get started with our pop quiz. Okay, why is everybody's camera off tonight, except for Nathan? You guys are here. Turn on your cameras if you can, folks. Here we go. That is the way to act like a class. There you go. Okay. No worries. You guys aren't trying to hurt anybody's feelings, but I don't want Nathan to feel like he's the only person with his camera on. Okay. Also, I do want to talk about real quick the tests. Majority of you did a great job on the test. That was awesome. I saw a lot of great grades. That was really encouraging. I did, like I did last time, write comments in the free response questions inside of the McGraw-Hill website. I have only graded half officially. I had to wait for parent emails, basically confirming proctoring. I want to make sure that that happens before I can give you an accurate grading. And I was just waiting on some parents to email me back. So those half, if you haven't gotten a grade yet, don't stress out. I plan on doing that by this weekend. I will get you the grades. I will hopefully get them tomorrow. So if you don't have a grade today, you will have a grade tomorrow. Do not stress out about that. And as usual, if you have any questions about your grades on the test, about your free response, I am not an easy grader. I don't claim to be. I also am not trying to be overly harsh. What I want to be is realistic. And hopefully you get that from the comments that I give you in the grades. I want you to be successful at the exam. And that doesn't mean coddling you and babying your experience in this class. It also doesn't mean being rude and not nice to you, because that's not helpful. And I wouldn't want to do that anyway, because I'm not that person. I want to be as realistic as I can in the comments. And so I may have some fairly long, detailed comments, but I want them to be helpful to you. And so please go back and make sure to read your comments and then respond to me if you have any questions about them. Nathan says, I haven't been able to view my comments for the free response questions. Okay, I will go back on the test settings and double check that everything is viewable. It should be. If it's not, I'm not sure why. So I will go back and find out. Thank you for letting me know, Nathan. When was the last time you checked?
Just now.
Just now. Okay, I will go back and check then. Thank you. Let me write a note to myself so I don't forget. Okay. Does anybody have any questions about the test before we move on?
I can't see any of my, couldn't see the worksheet. I don't know if that was because it wasn't fully finished. Like the entire score sheet wasn't I couldn't access it.
That's what Nathan just said. I just said I would check on it. So I will check on that for everybody and see if it's a test setting I need to switch. So sometimes it says you have to choose if and when students can view things. And so sometimes a setting there is a little bit wonky. Honestly, I really appreciate the website, and the website is really helpful as far as the settings that you can choose. A lot of it is just me figuring things out. And so sometimes there's not a test setting that's clicked right. And so that's my fault. I'll go and fix it. So I will check on that. Thank you, Orion, for the double checking that it's not just a setting on Nathan's. Cool. I'll go back and check. Anybody have any other thoughts or questions before we get started? In general, what I want to encourage everybody to do, this is something that I saw with a few folks, and I know that we don't talk about free response questions too much in class. We talk more about concepts than we talk about how to phrase them and how to critical think your way through the experimental design that's usually provided with a free response question. And I know that the AP Classroom ones are going to be more closely resembling those that are on the actual AP exam. They're going to have multiple questions per experimental design. It's not necessarily, here's this concept, explain something about it. That's more what the quarterly tests are going to look like. Excuse me. And so I understand that those are going to be a little bit more simple than the complex ones on AP Classroom. And so what I want to do is, in general, what I would recommend is make sure that you read the experimental design really well. And if you already do this and you feel good, then just tune me out. But I just wanted to make sure that I cover it at least once in class. Make sure you read through sentence by sentence. And something that I do when I read questions, it's not an age thing, it's not a maturity thing, it's just what works. read through 1 to 2 sentences at a time, talk through it to yourself, whether that's kind of mumbling to yourself, or if you can rewrite it, if you have extra paper, if you can think it through, if you have the ability to think in your head. Some people don't have a head voice. If you do, that's great and it's helpful. If you don't, write it down. Talk through sentence by sentence, make sure you understand, summarize it in a different way than what's actually written. Make sure you understand what's going on in the experimental design, what the purpose is. Always quiz yourself what the dependent and independent variables are. What is what's the control if they have one, or what could be a control? What are they trying to find out? And then make sure you understand fully the graphs, the data tables, things like that. Make sure to read them really well. That way, when you then look at the questions that they ask, you'll probably know what the answers are already because you've fully understood the experimental design. If you try to go the opposite way around, and if you read the questions first and try to cherry pick the results from it, some tests you can do that, some types of tests. With free response questions, you can't do that. You're going to have to extrapolate information for the most part from that experimental design. And so you need to make sure you understand it before you look into the questions. The other piece of advice I would have is make sure that you are as detailed as possible. Usually 1 sentence is not going to be enough. If you put a phrase, remember what they write at the top of AP Classroom, whenever you take the free response question kind of quizzes that I give you, they will say, outlines are not acceptable. Bullet points are not acceptable. Make sure you do it in a formal paragraph format. That's what they tell you on AP Classroom. And that's what they'll be expecting on the AP exam. So make sure that you take the tests that I give you in that same way. Always provide a reason for what you think. Always provide the reasoning, the logic, the evidence that you find. Sometimes they'll say based on the information above, or justify the claim that blank, or cite evidence to support blank. They're expecting you to go back and pull information and pull concepts from that experimental design and bring them into your answer. So if you get the right answer, that is going to be partial points, but make sure that you have the reasoning to support it to get full points. It's like in math class, if you didn't show your work, you may only get half credit. They want to make sure you understand why your answer is correct, not just that you picked something and it was right. So that would be my second piece of advice. Make sure you always give as much detail as possible. Always give too much detail rather than not enough. You should never have to write more than, I'd say, 3 small paragraphs, 2 large paragraphs. You should never have to write an answer above that. However, make sure that you give at least more than 1 sentence to support your reasoning, because if your answer is 1 sentence, you need to have several sentences of support to back it up. So that is my advice to you. Make sure you give detail. And fully read the questions. Sometimes I saw several students where they had put all of the answers for a free response question into the first fill in the blank, and then the rest of them were left blank. Even though all the answers were correct, they were piled into 1 blank at the top. And so that was kind of strange, but also some of them only answered the first question. So some of that is from free response in AP Classroom, some of that is from McGraw-Hill, but treat both the same way, give the same detail, make sure you read all the questions. So I will get off my soapbox unless anybody has any questions or thoughts. Multiple choice answers for the College Board unit checks. No, I'll go back and look at that. Which unit are you talking about in particular? Do you know? Is it unit 3 maybe? Or unit 4? 2 and 3. Okay, I'll go back. Cool. All right. Let's see. Let me make sure I haven't missed anything. Okay, you guys ready for a pop quiz before we start on our chapter? Okay, I will share my screen. And we will jump into the slideshow. You guys see what I'm showing?
I can see it.
Okay. So pop quiz question number 1. Why is meiosis so important? Why is it such a big deal? There can be multiple answers to this.
It allows sexual organisms to reproduce.
Allows for the production of gametes, which allows for production.
What was the last part? Production of what?
Allows for reproduction in the formation of a zygote.
Zygote, yeah. Some of these will be the same word in like several different formats. So there's gamete, there's gametophyte. All of it basically means like oocyte. There are a lot of prefixes and suffixes. You guys, every week we'll talk about prefixes and suffixes. A lot of that isn't super important. Zygote is referring to when the two gametes merge. So exactly. So it does help for the production of gametes, which then merge and become zygotes. The male and female come together. What else? What process makes it so important? Hang on just a sec.
Reproduction, sexual reproduction. If that's
Yeah. So I'm looking for something more conceptual than physical. What do you think is important about meiosis versus
It allows
mitosis?
for genetic variation.
Yes, that is a huge thing. That's why meiosis is so much different than mitosis. And what is the process that causes that to happen within meiosis?
Crossing over and independent assortment.
Absolutely. Crossing over independent assortment and the synapses that we'll talk about from this chapter. That's just another way of talking about it. So awesome. Next, when does recombination occur approximately? Give me a ballpark. Remember, recombination and crossing over are the same thing.
Isn't it prophase 1?
Yeah, I was going to agree with Nathan.
Prophase 1 of meiosis.
Prophase 1. It can be prophase 1 and it can go into metaphase. That whole process of them being close to each other, the homologs being close to each other, that whole time they can be switching genes back and forth. So absolutely. Beginning of meiosis 1, that was the most important part. So prophase 1, metaphase 1, prometaphase kind of, stuff like that. Okay, are the resulting daughter cells of meiosis 1 haploid or diploid?
Haploid.
Diploid, right? Meiosis 1.
It's haploid.
I have a haploid, I have a diploid. What do we think? What's the consensus? Anybody else?
Haploid.
Haploid? Okay, the answer is haploid, and we'll talk about why. So if you're not sure why, stay tuned, we'll talk about it, because there is a very confusing concept of actually what the word chromosome is, and haploid and diploid have to do with that, and so we'll give some clarifications. So don't stress, we'll talk about that. Which is also called trisomy 21.
Down syndrome.
Down syndrome.
Yes. Awesome. And we'll talk about what that is if you're not sure what trisomy is. Most of you guys, I'm sure, have read the chapter at this point. You guys know what we're talking about, but we'll go over it just in case. And then which presents as XO.
Is that Turner syndrome, I think?
Yep, exactly. And for bonus points, what is the presentation of someone with Turner syndrome?
Shorter.
Female.
Shorter female. And what happens as far as development, like physical development?
They don't experience puberty.
You said they don't experience puberty?
I'm pretty sure that was Turner syndrome. They have underdeveloped uterus and ovaries.
Yep, exactly. So Very interesting stuff. We'll talk about all of these in a little bit more detail later. So great job on those, you guys. Awesome. I feel like you guys are already prepared for this. Fabulous. Okay, so we're going to review mitosis real quickly first, and then we'll talk about meiosis, and then we'll compare them and how they're different. So first of all, a review. There's an image down below if you want to read that too. Prophase, DNA is going to condense into the chromosomes. The nucleolus is going to disappear, and the centrosomes are going to move apart towards the opposite poles. The spindle fibers are going to initiate their building. Prometaphase, which is kind of a new concept, but in between pro and metaphase, spindle fibers are going to be completed, and they're going to attach to the kinetochores of the chromatids. Remember, the kinetochores are the protein complex in the center of the chromatids. Metaphase is going to be the spindle fibers arranging the chromosomes along the center of the cell. They're going to be right next to each other, one chromosome, one chromosome, one chromosome, all the way across. Each one with two sister chromatids, right? Because that's what happened during synthesis of DNA in S phase. Anaphase is the spindle fibers separating the sister chromatids, pulling them toward the centrosomes. Depending on who you ask, anaphase or metaphase is quicker. I tend to believe anaphase is the quickest phase of the four. And then telophase or telophase, chromosomes are going to loosen back into euchromatin. I just put chromatin, but it's euchromatin. The nucleolus is going to reappear. The nuclear envelope is going to be rebuilt around all of it. And then the cleavage furrow in the center is going to be initiated. And then, of course, after that, we see cytokinesis, where the cells actually pinch apart. And then we go back into interphase. So that is mitosis. We talked about that last time. And the main thing that you need to know is that the result of mitosis is going to be two diploid daughter cells that have one sister chromatid each. As you can tell, before, when we're in S phase, the DNA is going to replicate each of those sister chromatids. That's two different copies of the DNA, one from mom, one from dad, is going to copy itself to become another sister chromatid. So instead of one red, we have two red, for example, one blue, two blue. And then those are going to divide during mitosis. They're going to split. And then one copy of each of mom and dad's DNA is going to go into those individual cells. So those are diploid, even though they don't have another sister chromatid to bind to the red and another sister chromatid to bind to the blue. And so let's talk about that. First of all, homologous is a word you'll see a lot, and the noun version of that, homologs. Those are two of the same chromosome, AKA a chromosome referring to a certain strand of DNA that holds the sequencing for certain genes, right? So two of the same chromosome, they just have different alleles, okay? They have two different versions or outcomes of the genes. So you could have one of them, one chromosome codes for red hair, one chromosome codes for brown hair, and those can be homologous chromosomes just because they're the same gene, but they hold different alleles. So we see these in diploid cells because each one of them is considered to be in. So when we have in, which is an understood one or one in, that means that we only have one copy of the genes. We either have moms or we have dads. We don't have both, but when we have mom and dads, even just one chromatid of each, that is considered diploid or diploid. So part of it is the confusion of the word chromosome. Chromosome can mean talking about a single chromatid, or it can be talking about when it's copied before mitosis or meiosis, the sister chromatid sets. The main thing is to figure out how many chromosomes you have, you count the centromeres. So if you look at the two karyotypes that are shown, remember karyotypes are a representation of all of the chromosomes in a cell, numbering from the largest at 1 in the top left to the bottom right being the sex chromosomes. The rest of them are autosomal. If you look at the left side, that is diploid or diploid. So they're not bound together. They are two different representations of the same genes in each chromosome. So each chromosome has two separate chromatids that are coding in different ways. However, right before you go into mitosis or meiosis, you're going to have what looks like the right, which is each of those chromatids having copied during S phase to become sister paired chromosomes. or sister paired chromatid chromosomes. So when you look at number 1 on the left, chromosome 1, that is diploid. There are 2 chromosomes. When you look at it on the right, there are 2 chromosomes. So the number of chromosomes haven't changed from left to right. The number of chromatids has changed. So both of these images are diploid. They both have 2 different copies of the same chromosome. One just has 2 chromatids of each copy. Okay. Does anybody have any questions about that? That can be very confusing and trip you up. Okay. If you come up with a question, let me know after class. Okay.
No,
So yeah, sure.
I had a question about homologs. How do you say it?
Homologous or homolog, depending on
Yeah,
what.
those. So those are. So this is the right. That's the image on the right. Those are 2.
So actually, okay. Sister chromatids, for example, if you look at what we call the X's, so on the right side, we have pairs of sister chromatids, right? Each of the sister chromatids in an individual chromosome are identical. It is copied. That is simply what it is on the left is what it is on the right. So that is not a homolog to itself. The chromosome, whichever one it is, moms or dads, if you look at those 2 X's in one on the right, one of those X's is going to be the homolog of the other X. That is also the first chromosome. They are coding for same genes, but they're coding for different alleles of those genes. And so if you look at the left side, if you look at the number 1 chromatids, aka chromosomes that don't have 2 sister chromatids apiece, they're just individual chromosomes. Those 2 chromatids, aka chromosomes, because it's still 2 chromosomes, are homologs to each other. They're not bound at the kinetochore, at the centromere, they're not bound. So those are 2 individual versions of that chromosome. So either way, on the left or on the right, the 2 chromatids on the left, the 2 larger doubled chromatids on the right, those are going to be homologs to each other. But sister chromatids that are paired in a single chromosome are not going to be homologs to each other because they're actually identical. So homologs have to be the same chromosome with the same genes, but different alleles of that chromosome. Does that make sense?
Perfectly not confusing.
Okay. I found a video that's actually very helpful to understand that. I'll link it to class after this in case you want to go back and listen to somebody else explain it in a different way. Would that be helpful?
Oh yeah, that would help.
Okay. I will upload that then. You should see it after class tonight. Okay. All right. So if we have, so remember the centromere is the center, the protein complex at the center of the chromosome. No matter if you have 1 sister chromatid or if it's copied after S phase and you have 2 bound together, you still only have 1 centromere. So what you can do when you look at images, when you look at diagrams, count the centromeres for each chromosome number meaning, and that will help you know if it's haploid or diploid. Okay. So moving forward, how do we apply that? So during mitosis, let's talk about synapsis and how that actually works. During mitosis, each chromosome, aka a pair of sister chromatids, so we're talking about after synthesis, so there are 2 chromatids, each chromosome is pulled apart during anaphase, right? So metaphase, they combine into the center in a line, and the equator of the cell. Anaphase, they're pulled apart, 1 chromatid each to each side. And so during meiosis, each pair of chromosomes is pulled apart. So not each chromatid, each pair of chromosomes. So we've got 2 full chromosomes, 4 chromatids, 2 and 2 are bound together. So we've got 2 full chromosomes, or 2 doubled chromosomes, you could say, if you're talking about the sister chromatids. Each pair is pulled apart. And what that causes is a synaptonemal complex, which is a very long word. I like to think of tetrad or a bivalent. Tetrad makes the most sense because you have 4 chromatids all basically laying on top of each other in the center of the cell. So instead of having one chromosome that's in the center and it gets pulled apart, each chromatid, you have 2 full chromosomes with 2 chromatids each. So you have 4 total chromatids in the center. And they're on top of each other, crossing over. So this is where crossing over occurs. They are going to be in such close contact that the bonds from one DNA strand are going to connect to the bonds of the other DNA strand in different places. And wherever they bind, they're going to cause the genes to interchange on the separate chromosomes. So this is what causes the recombination. And so the 2 full chromosomes, the 4 chromatids together, the 2 full chromosomes being together during prophase and metaphase 1, before they're pulled apart in anaphase 2, that is a long association time for each of those chromosomes to be able to recombine and cross over. So that whole point of synapsis is to get those 2 sets of chromosomes together to be able to recombine to cause genetic variation. So that's what synapsis is. That occurs in prophase 1 and metaphase 1. That's a big difference. So let's talk about meiosis 1 first. So listing it just like we do mitosis, it's similar except for a few differences. First of all, the homologs, remember that's 2 sets of chromosomes that have the same genes but different alleles. Homologs pair up for synapsis. That's what we just talked about. So those chromosomes, if you notice that image down at the bottom right, those are not individual chromatids. The red is a full 2 sister chromatid chromosome. The blue is a full 2 sister chromatid chromosome as well. So each of those sets of colors in the second image, you have 4 chromatids combined with spindle fibers. That is the crossing over, and that is what happens in prophase. And then in metaphase, you're going to line them up in the center. This is all while crossing over and synapsis is happening. Anaphase 1, you're going to, instead of pulling apart individual chromatids in a single chromosome, you're going to pull apart full double chromosomes with 2 sister chromatids apiece, but only after they have gone through synapsis and recombined. So if you'll notice in that third image, they have a little bit of blue on the red chromosome, a little bit of red on the blue chromosome. And so they have done their recombination. So they have plenty of genetic variation at this point. And this is meiosis 1. They still have plenty of dividing to do. So telophase 1 is kind of a little confusing when it comes to meiosis, because depending on who and which round of meiosis, sometimes it's partial, sometimes it's complete, depending on just the environment and the circumstances. I'm not exactly sure what goes into deciding how much telophase is done, but if you recall, telophase is the full division inside the 2 cells and the development of the cleavage furrow. All of that is done to prepare for cytokinesis. Sometimes cytokinesis isn't even fully done before meiosis starts over again. Meiosis 2 meaning. So interkinesis is what happens when you actually do full telophase and cytokinesis in meiosis 1. Interkinesis is going to be similar to another round of interphase in between meiosis 1 and 2. Sometimes it happens, sometimes it doesn't. You don't need to know in particular detail why, or obviously that's way beyond the scope of this course, but interkinesis is what happens after cytokinesis that is kind of like interphase before the cells head into meiosis 2. They sometimes do it, they sometimes don't. The difference between it and interphase is that there is no more DNA replication, there is no S phase, it's already done. They're just going to go straight into meiosis 2 where they will divide the DNA again. So it only needed to be copied once before meiosis starts. It doesn't need to be done again between meiosis 1 and 2. Otherwise, the cell is growing, more cytoplasm, more plasma membrane. They are continuing to grow. Interkinesis is very similar to that. Sometimes it happens, sometimes it doesn't. So then when you look at meiosis 2, you have same names, just number 2 after them. So prophase 2, the spindle fiber framework is going to get created again, similar to every other prophase. Metaphase, the chromosomes. Now the chromosomes are going to line up along the equator. So these are identical copies well, they were identical copies before recombination. Now, because of recombination, each chromatid is going to be unique. But hypothetically, if recombination hadn't occurred, this would look very similar to mitosis. So they're going to line up across the equator. Their sister chromatids and anaphase are going to separate them into daughter chromatids. That's going to look similar to what mitosis would be if you didn't have recombination. And then telophase 2 is the same as any other telophase, where the DNA unravels into euchromatin again. The nuclear envelope is going to be created, and then cytokinesis is going to happen next. So in meiosis 1, the interkinesis, referring to that telophase, sometimes it mentions in the textbook, sometimes the nuclear envelope doesn't even fully develop before they go ahead and go to meiosis 2. So this process is very quick, one after the other. Sometimes they don't even finish the process fully before they start dividing again into new cells. So it's just something to think about. Telophase 1 is kind of up in the air as far as how many cells actually do it. Okay, so now a comparison of mitosis and meiosis. And this, of course, is theoretically, and we'll talk about why it's theoretical in just a minute, but this is hypothetically what would happen in meiosis 1 and meiosis 2 as opposed to mitosis. So the differences between meiosis 1 and mitosis is that in meiosis 1, the homologous chromosomes are paired instead of single chromosomes lined along the equator of the cell. It also has synapsis in prophase 1 and into metaphase, where the tetrads are combined in the center of the cell and coming together even before they're lined up, and that allows for crossing over and recombination. So that is not in mitosis, because mitosis is a perfect copy. The chromosomes are separated. The homologous chromosomes are separated. One of the homologous chromosomes to one side, one of the homologous chromosomes to the other side, whereas mitosis is just chromatids from an individual chromosome. And then the result of meiosis 1 is 2 haploid daughter cells with 2 sister chromatids each. Now, why is it haploid? Can you tell me why it's haploid? If it has 2 sister chromatids bound together, why isn't it diploid? Anybody know? Okay. So 2 haploid daughter cells, haploid referring to the fact that there is 1 centromere. So you can count the centromere to know each of the chromosomes, even though they're recombined, they're still considered to be an individual chromosome. And so what's going to happen is, because they are bound at the centromere, they're considered to be 1. And so because of that, even though there's 2 chromatids, they're bound together, they're considered to be haploid. So what you were doing is hypothetically going from 46 chromosomes to 23 in each cell. You went from 1 cell with all of them to dividing 23 chromosomes into either cell. And so you went from 92 chromatids to 46 chromatids per cell. Okay, so you're dividing everything in half in meiosis 1. You went from diploid or diploid to haploid. In just a second, make sure I'm not missing anything. Okay. So meiosis 2 just has a little bit different. The haploid number of chromosomes along the equator, that is different, because when you think about mitosis, even though you don't have 2 homologous chromosomes right next to each other in synapsis like meiosis, you do in a regular mitosis. Mitosis is not a sex cell. Mitosis is a regular body cell. So your skin, your bones, your muscles, whatever that is, they all have 2 copies of DNA. They have your mom's and your dad's, whatever you got when you were born. So the difference is that they live in your cell normally as individual chromatids. They copy in S phase before mitosis, to where you then have full chromosomes of 2 sister chromatids apiece, but they aren't together. They're in individual places along the equator of the cell. So what happens is those full chromosomes that are individual, mom's is somewhere else, dad's is somewhere else, those get pulled apart to where they then have another individual chromatid, another individual chromatid from mom, one from dad, and then, but that's different than having a haploid number of chromosomes along the equator. They have a diploid, even if it becomes individual sister chromatids. So haploid is a little bit different than that. You start with haploid at the equator. That's different than mitosis. And then the result of meiosis 2 is because you started with haploid, but just doubled haploid, AKA 2 sister chromatids apiece, you now end up with 4 haploid with 1 sister chromatid apiece. So those haploid full chromosomes that had 2 sister chromatids apiece got split in meiosis 2. And so in meiosis 2, you go from those 23 chromosomes to still 23 chromosomes, but now you have 46 chromatids per cell to 23 chromatids per cell. So a little bit different. One of them is splitting in half and the other one is splitting in half, but a different way. Okay, so gamete formation, how is it actually different than the quote unquote theoretical? The production of sperm is different than the production of eggs. The production of sperm is basically a 1-to-4 ratio like you would expect in meiosis. 1 cell goes through meiosis 1, becomes 2 cells, goes through meiosis 2, becomes 4 cells. And so they have 23 chromosomes, only 1 copy of each. They are 1N or they're N, referring to haploid. And that is because they will be uniting with a different sex cell that has also 1 copy. When they come together, they form 2 copies of the DNA that are each different, and then that forms a full human. So 1 sperm to each egg is what's created, or 1 sperm to each final haploid daughter cell is what's created. Oogenesis, which is the production of eggs, is very different. So what you actually have happen is the initial diploid cell, very similar to spermatogenesis. You have an initial single cell that is 2N, it's been copied, it's ready for meiosis. It goes through meiosis 1, but then when it goes to split, one of the daughter cells takes much more of the organelles, the cytoplasm, the cytosol, much more than the other one. It totally dominates it in size. And as a result, they're not equal. They're really not. One of them is able to function a lot better than the other one. And so the one that is really small and weak and doesn't have a lot of cytosol, doesn't have a lot of organelles, is considered to be what we call a polar body. The one that is the winner, so to speak, the dominating one that has more cytosol and more plasma membrane and more organelles is considered to be an oocyte. It's the one that continues on in meiosis. And each time a polar body is created, sometimes if it's a more even 50-50 split, it can vary depending on the individual situation. But if it's closer to being the same size, then it may actually end up dividing again and becoming an egg. But it's not very often that it does that. A lot of times it becomes a polar body is what we call them. And what happens with that is it is usually dissolved or recycled into the cell, into basically the extracellular matrix to be able to be absorbed by molecules into other cells. So they will be recycled and used in other things, but it usually does not survive the process as a individual independent cell. So the big one that survives, the oocyte, actually goes through meiosis 2, and then it does the exact same thing. It creates a second polar body. And then the one that's left that is larger in size is called an ovum, which is what becomes an egg. So we, in oogenesis, during meiosis, produce 1 viable egg, usually, and 2 polar bodies. So 1 becomes 2, and then that becomes 2 again. So we really only have 3 products instead of the usual 4. And 2 of them are not viable, usually. So we have, instead of a 1-to-4, like the initial cell to sperm ratio is, because there's 1 daughter cell to 1 sperm, we have a 1-to-1 ratio in egg production. And you can see why, because look how large eggs are compared to sperm. There are a lot of sperm in that image that is 1 egg. So it actually, the process of fertilization takes more than 1 sperm. The sperm actually have to beat their way through the cell membrane, which is a very thick cell membrane, a tough one, they have to actually beat through and dig a hole into the membrane to be able to get through and actually fertilize and exchange genetic information. So it takes a whole village to fertilize one egg instead of just the, hypothetical one sperm that you think of. But eventually one final sperm will reach the genetic information, but it takes a team of them behind it to actually get through the tough exterior of the cell. So, but notice the size difference. That is because it is not an even meiosis. It has a small polar body produced each time, and one large cell that's produced at the end that becomes the actual egg. So with all of that interestingness, does anybody have any thoughts, any questions? Yes, it is a very confusing thing to say. Oocyte is what I've heard. You basically pronounce it twice, oocyte. If you want to say it differently, you are more than welcome to do that. Any questions? Okay. You guys are being awfully quiet tonight. Is that a good thing or a bad thing? Is this confusing, or are you just hanging out taking notes? Wi-Fi's lagging, or Ryan's hanging out. Okay. The reason I ask is not to put anybody on the spot. It's because I want to make sure I can cover something if it's confusing. Okay. And you guys know the slideshow will be up afterwards. Hopefully it will be helpful. Okay, I'll share my screen again and we'll keep on moving. Okay. Can you guys see my screen?
You can see it.
Okay. So we talked about meiosis, how it's different from mitosis. We talked about gamete formation. So hypothetically, you should produce 4 resulting cells, but in oocytes and oogenesis, you're going to produce really 1 viable cell. So that's why it's sometimes it works that way, sometimes it doesn't work that way. So something really important I want you to know, and that, of course, I look at College Board's course and exam description. The exclusion statement that they have for this part of the unit is the details of sexual reproduction cycles in various plants and animals are beyond the scope of the course and the AP exam. So we will not need to talk about pollination. We will not need to talk about individual types of sexual reproduction. You need to know mitosis and meiosis, how they're different, what the details are, how it relates to gametes that become zygotes. And that is the extent of the sexual reproduction that you will need to understand. Okay, so don't stress about pollination or anything else like that. We won't be studying anything other than this when it comes to sexual reproduction and cellular reproduction. Okay, so this is where it kind of gets interesting, talking about the application of genetics and the application of meiosis and what happens when things go wrong. So ploidy is referring to the number of copies of the gene, the number of alleles in the cell. And so usually you have euploidy. Don't know where did I write that. Okay, so before we talk about that, an actual ploidy, non-disjunction is a very long word. It is the improper separation of chromosomes. That means that instead of the spindle fibers working correctly and pulling one chromatid or one chromosome to each side, it pulls everything to one side by accident. The spindle fiber checkpoint doesn't get approved. So remember, after metaphase, before anaphase happens, there is a metaphase checkpoint. That checkpoint has proteins running all up and down the line of chromosomes, making sure that everything is connected properly. If it's not, they'll try to fix it. If they can't, they signal apoptosis. For this reason, if something happens and non-disjunction happens, in mitosis, it's not important because that individual cell will have a problem. And if they recognize that there's a problem, it's going to cause apoptosis and bam, the cell is gone. It's not going to cause that much of a problem. However, in germ cells, in gametes, and sex cells, you can use any of those interchangeably, non-disjunction is huge because you are a human that develops from one cell that meets with another cell. And if one of those 2 cells has genetic information that is messed up, then a lot of difficult things can happen for every single other cell after that. So if you meaning, a hypothetical you of nature, if nature messes up the initial gametes, then everything after that is going to get divided with that same problem. So meiosis non-disjunction is very important. So if you're confused about that term, I've highlighted 3 different sections of it. Remember, prefixes are huge. Non means no or not. Dis means away, junction. So if you look at disjunction, disjunction would be the separation, the pulling apart, the away from the meeting point. The junction is where they meet together. Dis would be away from that. So the pulling apart didn't happen correctly. So not pulling apart correctly would be the correct breaking down of that term, non-disjunction. So it's kind of like a double negative. So there are 2 types of errors when it comes to non-disjunction. It can happen either in meiosis 1 or it can happen in meiosis 2. And so meiosis 1, first of all, look at the normal, which is A. You're going to start with 2 chromosomes that have 2 chromatids each. You're going to separate the chromosomes. So 1 chromosome goes to one side, 1 chromosome goes to the other. Great. That's what usually happens in meiosis 1 after synapsis. And then those 2 divide in meiosis 2 and become 4 with all 1 copy of that gene, whatever it is, if we just hypothetically call it a gene. Each one of them has a different gene, and that's great. So then we look up at the secondary, which is if it's not as big of a deal because some of them might be normal. You have meiosis 1 goes normally, but then in meiosis 2, one of those preliminary daughter cells pulls the chromosome all to one side instead of dividing it into 2 chromatid chromosomes. The other daughter cell has divided correctly. So you go from 2 blue chromatids bound together to 2 cells with 2 chromatids, 1 in each. That's normal. But then you have a 50-50 chance of the gamete working correctly. The other one, because it happened in meiosis 2, has one that's too many and one that's not enough. And so those are going to have problems, but you have a 50-50 chance of it working right in second meiosis. However, when you have a non-disjunction issue in meiosis 1, you end up with no cells correct because you start out with all the chromosomes on one side and one cell has none of the chromosomes. And then when you divide those, nothing divided by 2 is also nothing. So each of those cells have nothing. And then the one that has both chromosomes divides to have 2 copies of each, which is not what it's supposed to have either. So primary, AKA when it happens in meiosis 1, is much more dangerous than when it happens in meiosis 2. So 2 important words to know is euploidy. Euploidy means that it is the correct number of chromosomes in the cell, whatever that is. So for us, it would be 2n. It would be diploid because the correct number of chromosomes that we should have when we have regular body cells is 2n. The correct number of chromosomes for our gametes should be 1n, right? Or just n, we leave off the 1, it's assumed. Aneuploidy, again, an means not or against. Aneuploidy is the incorrect number of chromosomes. It can either be too many or too few, caused by non-disjunction where things separate incorrectly. And so 2 versions of aneuploidy are monosomy, which is diploid 2n minus 1. It's 1 less chromosome than should be there. And the other option is if you have too many, you have trisomy, tri meaning 3, mono meaning 1. Trisomy is 1 extra chromosome that should not be there. So those are the most likely when it comes to people who have diploid cells to begin with. That's really your only options. Either one can be fatal. All monosomy is basically fatal. Trisomy has just a few that are not fatal. Trisomy 21 is really the only one that's rarely fatal. Trisomy 13 and 18, most people die before the age of 1. Too many physical damage to their physical development. They have holes in their heart, they have facial deformities, they have brain deformities, heart failure, things like that. It's really, really difficult. So, but we'll talk about that kind of stuff. And first of all, these are autosomal chromosomes. So everything 1 through 22 is autosomal. The 23rd pair is going to be your XX or your XY. So we'll talk about those in a minute. These are autosomal. These are the non-sex chromosomes. So first of all, trisomy 13 is patau. And so what you need to know is that if you have an error with your chromosomes, if you have 1 more or 1 fewer or a large dislocation or a problem with the chromosome, the lower the number, the bigger the chromosome is, and the worse of a time it's going to have because it holds more genes. So the lower the number, like chromosome 1 has a lot of genes on it compared to chromosome 21. And if you have a problem with chromosome 1, you are not going to survive. 21, however, it has so many fewer chromosomes or so many fewer genes that it really doesn't cause as many problems. It does obviously show, and it does have some physical issues that come along with it, but it's not nearly as deadly as an issue with chromosome 1. It even relates to chromosomes 13, 18, and 21. So 13 is the most severe of these three. They usually die before their first birthday. They have massive physical deformities in the face, heart issues, head deformities. They have microcephaly, things like that. Microcephaly, micro meaning small, cephaly meaning head. They have very small heads and very small brains, and are not able to function as well as they need to. So they usually die. Some don't, but some survive, but it's a very difficult life. A lot of surgeries, a lot of medicine. Trisomy 18 is still pretty severe. Edwards syndrome is what that's called. This person usually dies before their first birthday as well. They have severe physical deformities. It's hard to survive that. But then trisomy 21, because it's much smaller of a chromosome, is Down syndrome. You probably know somebody who has Down syndrome. It's an extra 21st chromosome, trisomy, so tri meaning 3. And the physical development of that person is altered. So they usually have some pretty defining physical characteristics that you could probably kind of imagine in your head if you've ever met somebody with Down syndrome. They have intellectual disabilities that are usually present. Sometimes they're more severe than others. A lot of people with Down syndrome can work regular jobs and go to school and go to college and many things like that. They do have usually some sort of mental disabilities, but some are more serious than others. They usually also have heart problems because their hearts aren't built fully. They're either underdeveloped or they're developed wrong. A lot of people have holes in their heart. With a lot of these, they don't develop correctly. So trisomy 21 still has some issues, a lot of physical problems that they have to deal with medically for the rest of their life, but they can survive, and a lot of them have wonderful, happy lives, and that's awesome. Especially as we had more research on heart surgery and things like that, we have been able to actually fix some of the issues caused by Down syndrome. So that's been pretty cool. So then if you look at sex chromosomes and how those are different, if you have disjunction with sex chromosomes, first of all, if you have only one chromosome, if you have only one Y, if you have no X, you will die. You cannot survive without an X. Everybody's got to have an X. However, you can survive without a second chromosome if you have an X. So that is XO, like we talked about in the quiz, that is Turner syndrome. A lot of people actually have Turner syndrome. They may not even know it. It's a pretty common thing, but just a little bit of physical underdevelopment. They only have one chromosome, the O. Whenever you see an O, when it is referring to sex chromosomes, that is representing the absence of a second one. There's not actually an O chromosome. So I'm not sure if we talked about this in class yet, but when you have two X's in your chromosomes, if you're a female or if you're a male with Klinefelters, which you have two X's as a Klinefelter, you have one of them shut off. And I say shut off a little bit loosely because they actually do use it for some things. We're still actually doing research as to what we use that deactivated X for. But for the most part, most people really only need one X, except for a little bit of function from the second one. And so the one that's deactivated is called a Barr body. And the Barr body is actually what helps us go through puberty, and females referring to. So if a female does not have a second X, they don't have a Barr body anywhere in their body. And so they're going to be physically underdeveloped. They won't go through traditional puberty. They will have underdeveloped ovaries, but as a result, they're not going to be able to have their own children, but with donor eggs and in vitro fertilization, they can have children. So it's a little bit different, but with hormone supplements, they can lead fairly normal lives. But that is someone who has only one chromosome, sex chromosome in their cells. So then what about the opposite of that? What if you have two X's and also a Y? That's Klinefelter. Klinefelter is an extra X chromosome, and that deactivates as it does in females, and it becomes a Barr body. And that causes men to share some female characteristics. It can cause a puberty similarly to females' puberty because of the bar body having an effect on that. And so they will have underdeveloped reproductive organs. They usually are sterile, and they have a minor to major amount of breast development because of going through partial female puberty. That is a fairly common one as well. Klinefelter is actually also the reason why some calico cats are male. It's very rare to meet a male calico cat. I've met one in my life. However, it's because bar bodies play into the coloring of a calico cat. The coloring of a cat's fur is actually on their sex chromosomes, and so partially at least. And so whenever the bar body turns off one X or the other X in each of their cells, it determines what their color of fur is. And so the earlier in their development in utero, the earlier that those bar bodies are deactivated, the bigger the splotches of color will be, because anything that came from that original cell that deactivated the bar body, any other cells that come from that in mitosis in the womb are going to be that same color and have that same deactivated X chromosome. So calico cats are most of the time female because it requires 2 X's and a bar body. But if a cat has Klinefelters, it can actually have calico fur as well. So that's funny and interesting to research if you're interested. Another thing is poly-X, or the super female. That is when you have an extra bar body or more than one extra bar body. If you just have an extra one, that's going to be someone who's tall and thin. Otherwise, not really any difference. Potentially intellectual disabilities, but not usually. If you have 2 or more extra bar bodies, then they are also taller in size, but they are going to have severe intellectual disabilities. If you have that much extra chromosome information, you're going to have some issues. All of these people are usually normally fertile. They can have children. That doesn't really, because they have the X that allows them to go through puberty, nothing else is really affected. And then the last one is, what if a male has 2 Ys? So they have XYY, they have 3 chromosomes, and that is Jacobs syndrome, which is a taller person than average. Usually they're plagued with a lot of acne, which I think is very interesting. And they have speech and reading problems traditionally. Otherwise, they can have children. They physically look normal. They're just a little bit taller than average, and they struggle with acne. So those are some more common sex chromosome disjunction syndromes. Hopefully you find those interesting like I did. So I'm going to go ahead and see if we've got any questions. Everybody doing okay so far? Okay, you guys, I'm speeding through this because it's a lot of information. Hopefully it's all making sense from what you read in the textbook. Okay, one more time. All right. Let me know when you guys can see.
I can see.
Okay, thank you. So we've talked about non-disjunction, which is when you have an extra chromosome or too few chromosomes, but what about when the chromosome itself has a structural change? So that's a little bit different. So there are different types, and I've listed them here. This is not referring to mutations and things like that. This is a physical issue with the structure. Mutations and things like that, if something gets attached wrong by accident, like a point mutation, that's not what we're talking about. This is the larger things that are referring to basically whole genes. So when you see segment, think at least one gene. So deletion is when a segment of a chromosome is removed. That is just gone, but there it goes. That can cause extreme problems. We'll list them below. Duplication is where an actual segment, a gene or more, is duplicated. That can cause different issues as well. Inversion is when a segment is flipped on the chromosome, so it goes backwards 180 degrees. Translocation is when a segment is transferred to another chromosome that is not homologous. So something from chromosome 5 goes to chromosome 21. That can cause all sorts of issues. So the most common things, and feel free to memorize these, but you don't need to memorize every single syndrome ever. These are the most common that you'll see when it comes to syndromes from changes in the structure of chromosomes. Williams is the first one where chromosome 7 loses the end piece. And so that's going to be not very fatal. It's usually going to be abnormal facial features. They have kind of a pushed up nose into their head. They have differently placed ears, and just kind of things that you would notice kind of look a little bit different than the usual human appearance, but nothing that's damaging. They are usually lacking in academic knowledge and academic interest, kind of intellectual development, but they are above average in expression types of abilities like music and speaking and verbal stuff. So that's just how their brain is developed a little bit differently. And they have the curse of aging quicker because one of the proteins, elastin, which is involved with your skin elasticity, interesting from the word, you would probably get that, has to do with a protein imbalance. They can't produce elastin the same way. And so because of that, their skin ages quicker than usual. So that is something interesting with Williams. Cri du chat is the other one, which is French for cry of the cat. And that is where chromosome 5 loses its end segment. So remember, the smaller the number, the more damaging the problem could probably be. So chromosome number 5 loses its end segment. That kid is going to have microcephaly, which is small head, intellectual disabilities, potentially facial abnormalities, and it actually causes your larynx, your talk box, so to speak, to be developed abnormally. And it causes the child, when right after they're born, when they're young, to cry like the meow of a cat. So that is why it was named Cri du chat. Okay, so does anybody have any questions before we go to our discussion? Yes, one
None
last
from
time.
me.
Say that again.
None from me.
None from you. Okay. Gonna wait another second in case anyone's typing. Okay, so without further ado, I'm going to hand it over to Ramaisa, who's going to be talking about our chapter this evening, and hopefully asking some very interesting questions that we'll all be able to talk about. So Ramaisa, you want to take it away?
Sure. Should I put the questions in the chat? Or?
That would be fabulous. I think that's worked pretty well.
Okay.
Thank you.
All right, the first question is, how many stages or phases are there in meiosis, and what are they?
There are 8 or 9 technically, I guess.
Okay.
If I'm counting, like, if we're counting prophase 1, metaphase 1, anaphase 1, telophase 1, interkinesis, and then prophase, metaphase, anaphase, and telophase 2.
Yeah. That would be
And so-
9, including interkinesis.
Yeah.
After telophase 2, is there cytokinesis? Or is it
Yeah.
all one step?
No, there is cytokinesis, but then the question is, if cytokinesis isn't part of mitosis, is it part of meiosis? There's the
Uh-oh.
question. What do you
Okay,
think?
it's not.
Opinions? No? So then it's like, well, is interkinesis a part of it? I guess kind of, because it's kind of in the middle. So there could be different answers to that, but I think that's what I would go with 9. Plus 9 plus 1.
All right, the next question. Okay, so can we summarize like the things that happen in meiosis? Here. I added this kind of in question 1, so it's like summarize little things that happen in meiosis.
There are different answers for this.
A diploid cell becomes a genetically unique, or it turns into 4 genetically unique haploid cells.
That works.
That was a very good summary, just bookending the whole process. Love it. And that is the important thing. Knowing haploid and diploid is the kind of stuff that they're going to try and trip you up on. So it's good to know those kinds of things for the test. Awesome. What else happens with meiosis? So that's the goal, that's the end result. What's the purpose of meiosis? What's the point?
To mix genes to make sure that mutations are possible, natural selection and evolution can happen to ensure the existence of a species continues.
Did I tell you guys in class about the bananas? Do you know what I'm talking about?
No.
No. Okay. If I did tell you and you remember, feel free to stop me. I watched this very interesting TED-based video. You guys know TEDx kind of stuff. They have TED Ed, which is educational videos that are animated and very interesting. My husband found one that was about bananas, the dark history of bananas, which I found was fascinating. He didn't know bananas had such bad history, but the point of it was talking about around the late 1800s, I think, once transportation across countries had gotten a lot easier and quicker via big ships in the Panama Canal and things like that. 1800s going into the 1900s. Bananas were a huge thing. Everybody wanted bananas, and they were a huge hot commodity, which we don't really think of that now as just grab a banana, you know, but they were a hot commodity. And so big companies decided to buy for very cheap large swaths of rainforest in South and Central America to plant banana plantations. And all of these bananas got produced. All of these bananas, they were all one species, one variety, and they planted millions of acres of bananas. And they were transporting them everywhere, especially to America and Europe, to make a lot of money off of them, right? Except because there was no genetic variation, one disease came through and killed every single tree in like a year. And so they realized from that they should probably have some genetic variation in their bananas. And so they created a hybrid called the Cavendish, which is what we eat today. That is our usual banana. So that was you can kind of think of it as genetically modified, but it was basically just a hybrid between different types of bananas that had different resistances to different pests and things like that. So they encouraged not just producing clones of one plant, which is what they were doing, but they encouraged the free pollination to have that meiosis happen. That genetic variation, that recombination, that crossing over that produces the genetic variation that means that the whole banana plant, millions of hectares, don't get knocked out with one single disease. So there's an application of why meiosis is very important. You don't want to kill all the bananas because bananas are very important. So anyway, there's my story for you. Keep on going.
Do we move on to the next question?
Sure.
Okay. So the next question is, we went over a little bit of mitosis over class. So what are some of the differences between meiosis and mitosis?
In mitosis, the daughter cells are genetically identical, and in meiosis, they're genetically unique.
Okay.
What about the process? What parts of the process are different?
In meiosis, there's crossing over and independent assortment, and in mitosis, there's not.
So the law of independent assortment, I want to make sure that we're on the same page, that I know what you're talking about. So the law of independent assortment is talking about how the closer two genes are to each other, the more likely they're going to get crossed over together. So linked genes is what we call them, the closer ones together. So if gene 1 and gene 2 are right next to each other, in crossing over, if a big section moves, chances are they're going to go together. more times than not. And so that's how they were able to map out roughly the chromosomes and where the genes were, because they could figure out how often tall went with brown hair, right? So they looked at those overall genes or overall qualities, those phenotypes, and they could figure out from that how close together those genes were. So the law of independent assortment is talking about how far away the genes are related to each other and how that relates directly to how often that they are kept together. Does that make sense? Okay. Yeah, I just wanted to make sure that that was what we were talking about. Cool. Keep going.
All right. Then the next question is, what might happen to a damaged cells, or only one type of or if there was only one type of chromosome in meiosis?
What do you mean by one type of chromosome?
Like if there was only one particular one, there wasn't both the daughter chromosome.
So if it didn't properly synthesize a new copy before meiosis, is that what you mean?
Yes.
I got you. What do you guys think?
One or two of the daughter cells may have aneuploidy.
You think it would even go through fully meiosis too, if it only had one copy? What do you think? This is speculation.
I don't know.
Yeah, I thought it might not finish because you need both of them to go through.
Although it talks about in here, hypothetically, if you have non-disjunction, something with none of that chromosome would still divide. But that was referring to one individual chromosome, not a whole set of chromosomes. If you were haploid in everything, that would probably cause a little bit more of an issue than just non-disjunction of one cell or one chromosome. Interesting. What about if the DNA was damaged?
If the DNA was damaged during meiosis, then depends if it was after the checkpoint, then there could be a deformity in all of the resulting cells. There could be, the cell could just have apoptosis because something went wrong in cell processes, or it could just die eventually and not go anywhere.
And that brings up a question, which I will find out and link potentially an article, whether there are checkpoints in meiosis the same way there is in mitosis. Does anybody know? Because it doesn't mention in the textbook, so I will find out, because that would partially answer your question, Raisa. If there is a checkpoint, and if you can tell that the cell has damaged DNA, well, then it's probably not going to continue. It's going to have apoptosis go on. So I will find out for us.
All right, thank you.
Mhm.
I have one more question. Oh no, I have two more. Okay, so next question is, why must cells go through meiosis and meiosis 1 and meiosis 2? And what might happen to the cells if they only went through meiosis 1? Kind of similar to the last one, spreading apoptosis.
Through meiosis 1. What do you- I think there would definitely be a large instance of aneuploidy. Because if you ended up going through only one level for some reason, then you're going to have 2 copies of everything. And when it combines with the other gamete, that would be 3 copies of everything. So that would probably not survive. I don't think that would be a viable zygote.
Because meiosis 2 separates sister chromatids. So that
Mhm.
when fertilization occurs, it's one sister chromatid from the dad and one sister chromatid from the mom to make a chromosome.
Well, so they don't actually combine because chromosomes that are 2 sister chromatids bound together, except for recombination, which is kind of forcing them to be different. Other than that, those sister chromatids are going to be identical. So what happens is those individual chromatids, mom's copy and dad's copy, are going to unravel into euchromatin and just be in the cell. And then when it comes to mitosis, they're going to double and have sister chromatids copied of each of them. So then they're going to be full chromosomes, right? And then they're going to split those, causing individual chromatids again. So they're never going to bind together, mom and dad's. They're not going to make the full chromosome. They're going to be individual chromosomes because they're 2 different copies of that DNA. Or not copies, but 2 different versions of that DNA, one from mom, one from dad. So then in S phase, they're going to become chromosomes, full chromosomes that are double chromatids, right? Then they're going to divide those again into, I'm trying to do a gang sign, two different sets of chromatids. So does that make sense? So they really don't combine. They stay in the cell as individual chromatids. Because if they did combine, when they went through S phase, they wouldn't need to because they would already be combined. They would already have a sister chromatid bound. So in order to allow for S phase for mitosis, you actually have to have them separate. Does that make sense? Was that more confusing?
No, that makes sense.
Okay.
Then the last question. All right, so what are some things that make prophase in meiosis different from prophase in mitosis?
That's a good question. I love the youngins yelling in the background.
Sorry.
Oh, good.
But
If my microphone was more sensitive, you'd hear my husband yelling on the computer. He's playing with his friends. Okay. What do you guys think? What are the differences between prophase 1 and prophase?
Prophase 1 doesn't separate the actual chromatids, it separates the sisters. Or no, that's anaphase. Never mind, I'm wrong.
You were getting somewhere though. You have a point to that. Just think about how it relates to prophase. What happens during prophase 1?
Crossing over happens.
Crossing over. And that really all important S word that we learned this week, which is what?
Synapses.
There you go, synapsis. So during the process of the synapsis, the crossing over occurs of the 2 chromosomes. Each of these is a chromatid. That's why I do my fingers like this. The crossing over occurs, the 2 chromosomes. Things interchange in recombination while they're crossed over. So see, these words are related, but they don't mean the exact same thing. So during synapsis, the crossing over occurs because they're so close together. Recombination happens. And then metaphase happens, causing them to line up. And you have them lined up like this instead of like individual chromosomes, right? the two chromosomes are right next to each other. And so what happens in anaphase 1? That's what you were talking about. No, Orion. What happens in anaphase 1? Okay, Orion might have taken a bathroom break. Two chromosomes, two double chromosomes pull apart. So that's the first thing that happens in meiosis, anaphase 1. Then in anaphase 2, these are going to get separated. But the chromosomes, the sets of sister chromatids, get pulled apart first after recombination. Okay. Ah, yes, Orion was away. Okay, anybody have any other questions or thoughts? Thank you, Ramisa. Those were great questions. Okay. You guys look pooped. All right, well, if you want to, yes. Thank you. Those were great questions. You had a lot of questions. That was awesome. If you guys would like to stay and ask any questions, feel free. I will be here. Otherwise, I think we're good to go. I do want to share real quick. So by now you should have reviewed chapter 9. You should have read chapter 10. You should have done, preferably watched all of the videos, the crash course, should have done the AP classroom video on meiosis. I had 1 on mitosis versus meiosis, I think 2 on meiosis, something like that. And then preferably by Sunday night, please finish your smart book for chapter 10 and then do your meiosis lab that involves a worksheet from eMind. So if you forgot how to use eMind or need your password reset, let me know. Otherwise, please upload those to Moodle when you're done with the worksheet. And then I will grade them. And I don't know if everybody was in here when I said I will finish grading tests and get you your grade back at least by tomorrow night. Okay. And I will figure out the settings, Nathan, Orion, Noah, those of you who have mentioned that. I'll get you the ability to view them. So, and then I will add the video about ploidy, haploid, diploid, chromosome numbers, stuff like that. It was a really good video. I think Bozeman Science. And then I think there might have been one by the Amoeba sisters too. That was a good one. And then I will find an article referring to checkpoints in meiosis. So those are my assignments. Is that everything? Is there anything I forgot? Okay. All right. Then you guys are good to go. Thank you so much for being here tonight. Hope you do well on your homework, and I will see you guys next week. Have a good evening.
Thank you.
Thank you, you too.
Thank you. Bye. Good night.